Hi and welcome to the Controlled Experiments video. Please take notes while you watch. During the video, you should pick up a few new vocab words about controlled experiments. You'll learn about the parts of a controlled experiment and why we use them, and why it's such a powerful tool used in science. When you think of a controlled experiment, you probably think of a scientist in a lab coat mixing a bunch of stuff together to see what happens, and if something happens, then bang, you've discovered something. But unfortunately, it's not usually that simple, and it takes a little more work. And oftentimes, to definitively say that we have discovered something new, scientists must have completed lots of controlled experiments that all lead to the same conclusion. Before beginning a controlled experiment, it's useful to have a hypothesis. A hypothesis is a prediction based on observations, facts, or knowledge of how the world works. You will learn a lot more about how to write a hypothesis another time, but for now, let's just keep it basic. Essentially, it's an educated guess about how the experiment will turn out and why. A good hypothesis usually includes the three words, if, then, because. For example, I could say this hypothesis: if I breed a cat and a chicken, then I will get a chicken-cat hybrid because I know that breeding a donkey and a horse produce a mule. Sometimes one's hypothesis turns out to be wrong, which is all right. The goal of science is not to be right all the time. It's to learn new things about the world around us. For example, Thomas Edison, the inventor of the light bulb and many other things, made hundreds of prototypes of the light bulb that bonked. Good thing he was a persistent guy. He's quoted in saying, "I have not failed. I've just found 10,000 ways that won't work." Let's say we want to study plant growth. I could write a hypothesis for this experiment like this. If a plant is given more water, then the plant will grow taller because plants need water to grow. It's a really basic hypothesis, but it'll do. Please note that both of my examples for a hypothesis use these three words: if, then, because. All right, let's move on to the second part of the controlled experiments, defining our variables. How many things can you name that help plants grow? Pause the video for a second and write down as many things as you can think of that help plants grow. Each one of the things that you wrote down is a variable. We can change how much of each of these the plant receives. A variable is something that can change during an experiment. For example, the height of the plant may change, the amount of water, sunlight, soil, nutrients, oxygen, the space that a plant is given in the pot. All of these things are variables that affect plant growth. In a controlled experiment, we purposefully change only one variable to measure how it will affect another. In this imaginary experiment, we're only going to change the amount of water given to the plants to see how tall the plant will grow. We change the amount of water, we measure how it affects the height of the plants. Those are the only two variables that will change during this experiment, since we're going to try, to the best of our ability, to keep everything else that could affect plant growth the same. Let's look at our variables in a little more detail. Which is the variable that we're purposefully going to change during this experiment? Since we can't control how tall the plant will grow, that's not it. We do have control of how much water the plants are given, though. That's our independent variable. It's the cause of the difference in the height of plants. You can see the different amounts of water that each plant is given below each pot. We have varied the amount of water that each plant receives. Each plant is its own experimental group. The dependent variable, that's the variable that changes in response to or is affected by the independent variable. It's the effect. The height of the plant depends on the amount of water they are given. You can see the amount of water here underneath the plants. To make sure our results are reliable, scientists may run many trials of the experiment. A trial is just a repetition of the experiment. We should probably do many trials within each of these groups. For example, maybe we give 5 different pots 0.2 liters, and 5 pots 0.4 liters, and 5 pots 0.6, etc. I'm going to average out all the 0.6 liter pots because that'll give me a more accurate measurement of how water affected the height of the plant. So what happens to the rest of the variables we talked about, like type of soil, amount of sunlight, the amount of CO2 that the plant gets, the shape of the pot? Wind could affect the plant growth, temperature. What happens to all those? We can't know exactly how water affects plant growth unless we keep everything else the same during our experiment. We call the rest of these variables constants. To help you remember that constants don't change during an experiment, here are some synonyms for the word constant. Same, unchanging, regular, steady. This is a super important part of controlled experiments because without it, we'll never know how the results were affected by our independent variable, in this case the water, versus any other little change that might have occurred across our trials. The last concept for the day: lots of experiments use a control group as well as many experimental groups to determine the effect of the independent variable on the results. For example, let's say we wanted to test various types of energy drinks on students' concentration or performance in school. We might try these 3 different energy drinks. In order to determine what the normal level of concentration is for students, we would have a group that gets no energy drink. Sad for them. Their level of concentration or performance is considered our baseline, and that's what we're going to compare the concentration levels of the students who did receive the energy drinks to. The kids that got no treatment are our control group. The students that did get a treatment, or the energy drink, are our experimental groups. Having a control group is a really important part of medical trials for pharmaceuticals or new medical procedures. Keep in mind that there is not a control group for every experiment. There's only a control group if there's such a thing as a normal or untreated condition. Well, that's it for today. I hope you learned a lot, and have a good one.